Good morning, everyone. I would like to start by thanking the organizers for inviting me and putting together this nice conference. Not long ago, I was also a student, and we used to organize such conferences, inviting speakers, and it was very amazing to interact with the speakers as well as the many participants from many places in Germany. So, and of course, welcome to Caltech. I tried to show the... Okay, first let me get used to the... perfect. So I tried to show our beautiful campus here, and those of you who are not from Caltech, I hope you'll find some time to walk around a little bit and enjoy the campus. I will talk today briefly about most of the work I did during my grad school and also postdoc before starting here in Caltech 4 months ago. And it's on transcription regulation, specifically focusing on termination of RNA polymerases. So let's get started. Briefly, I actually come from Ghana on the west coast of Africa, so I was born in the very south of the country, grew up in a small town called Adan, and then moved to Accra to the University of Ghana, where I did my bachelor's degree in biochemistry. And I always wanted to do science at a very high level, so I was really grateful for the opportunity to get a position in one of the International Max Planck research schools in Germany, where I studied molecular biology, and there I actually fell in love with cryo-electron microscopy and structural biology in general because of the high level of information that we can get into biological processes when we apply these techniques. And so the key question that I'm interested in is in the first step of the central dogma, which is basically the information that drives life. It's stored in the DNA molecules that's contained in our cells. And this information is basically accessed through several layers of regulation. The first one being transcription, where RNA polymerases can find genes and are able to transcribe that information into an mRNA intermediate, which eventually becomes the blueprint for protein synthesis. And proteins in the end, as we know, are the workhorse of the cell and perform most of the functions in the cell. In eukaryotic organisms, 3 RNA polymerases are performing this role, called polymerase 1, 2, and 3. And all of them are very large machines that bind very tightly to their DNA template. And I will show you examples in the next few slides. In particular, RNA polymerase 2 is a central enzyme that transcribes all protein-coding genes and the majority of non-coding genes. And so POR2 activity is what really drives cellular processes from the very development of our cells into organisms, as we see, and also the eventual response of matured organisms like us to our environment, such as temperature changes, invading parasites, and so on. So POR2 activity is really at the core of everything that happens within us as we navigate through our environments. Now, okay. Now, our genes are organized in a way that there is the start of the gene called a promoter region. There is a gene body which is usually densely packed with what is called nucleosomes. And then at the end, we have the termination site. So even though we have a very long stretch of DNA on each chromosome, that unit is divided into these functional units called transcription units. And each of these transcription units is what encodes a particular gene. For example, your favorite protein has such a defined unit. RNA polymerases basically must find the start of this gene to be able to precisely synthesize the message that will encode the protein. And this is facilitated by transcription factors that specifically bind at the start of the gene. They then help the recruitment of RNA polymerase 2, which is shown here with this gray cartoon. POR2, with the assistance of these transcription factors, can then melt the DNA double helix and start synthesis of RNA molecule. As soon as the RNA molecule emerges from the polymerase, it is bound by a transcription elongation factor shown here as green. And these factors provide an enormous stability to the polymerase that allows it to transcribe to several bases of DNA. Imagine a gene that is 2,000 or more nucleotides. And sometimes this requires up to 13 hours to synthesize one gene. So you need a very, very incredibly stable machinery that can navigate that complex environment from start to the end. At the end of the gene, however, endonucleases find the end of the mRNA, cleaves it, and facilitate release of the polymerase from the DNA template. And this step is also very important because you don't want the polymerase transcribing into the neighboring genes that are not needed. And so there is very detailed regulatory mechanisms in place to ensure that genes are expressed in a timely way, in a precise way, including post-translational modifications such as phosphorylation of the C-terminal domain of RNA polymerase II, which facilitates the transition between this initiation phase, elongation phase, and also at the termination phase. Now, in higher organisms, there is another layer of regulation called promoter proximal pausing, which is a checkpoint in which shortly after the exit from the promoter, the RNA polymerase is held in a pause state, which is an opportunity to decide whether you want to synthesize the gene or not. So the initiation is not the end. There is another opportunity to basically change. The cell can start saying, okay, I don't actually need this transcript anymore, and then can stop the process here. Promoter proximal pausing is orchestrated by two elongation factors, the DRB sensitivity inducing factor DSIF and the negative elongation factor NELF, that bind the polymerase within this promoter region. And so when people started mapping the distribution of RNA polymerases across the gene, it became clear that most of them are actually held in this pause state here. And beyond this point, mostly the polymerase runs very fast through the gene body. Now, while many in vivo studies have allowed us to gain a lot of information on this process, recent development in cryo-electron microscopy has actually allowed us to visualize to the atomic level how promoter proximal pausing is orchestrated, and also how an activated elongation complex looks like. And I will focus briefly on the structure of the pause elongation complex. And so here is on the gray big surface is RNA polymerase II molecule. Nucleic acids are bound inside the cleft of the polymerase, and the elongation factor DSIF is the green molecule here, which basically binds the nucleic acids and traps them inside the polymerase, essentially sitting on the so-called POR2 clamp, locking the DNA inside the POR2 active site. And this basically prevents dissociation of the polymerase from the DNA template. NELF, on the other hand, binds the so-called funnel region of the polymerase and thereby tilts the nucleic acid in the active site so that the polymerase cannot actually progress anymore. And so at the end, you have an incredibly stable complex at the promoter proximal region that needs to be released into the gene body. And as I mentioned briefly initially, release of the pause polymerase requires a kinase that phosphorylates the elongation factors, the polymerase itself, and facilitates the exchange of these pausing factors for positive transcription elongation factors here. Now, the original idea when people discovered promoter proximal pausing was that most polymerases are held in this poised state and wait for a signal to be released. But once people started quantifying the duration of pause polymerases, it became clear that actually it's in the order of minutes. And so polymerases that pause don't necessarily have to be released. They don't sit there waiting for that signal. If there is no signal, they are immediately terminated. In fact, some of the recent data suggests that there is this rapid turnover where initiation happens and then immediately terminated, which has the advantage that you always have polymerases recruited, and if you have to respond to a signal, you quickly just prevent the termination and then can just progress into the gene body. Now, one of the factors that has been described to be key for that termination process is the integrator complex, which I will focus on for the rest of the slides. We know that integrator terminates transcription because it has an endonuclease. And when you deplete that endonuclease in cells, you see that there will be more polymerases accumulating in the promoter proximal region, and some of them getting released aberrantly into the gene body. And so how does the integrator function? Before I get to that, I want to quickly show you that this is a very large machine one of the largest in transcription, about 1.5 megadalton, has 15 subunits. Most subunits are really large, but the two things that I want to highlight on this slide is the similarity that was very apparent when we compare the sequences of Integrator to the human or eukaryotic proteome. 3 of the subunits share this methyl beta-lactamase and beta-caps folds that are unique to RNA cleaving enzymes. And so this was how we knew in the beginning that Integrator is an RNA metabolism enzyme. Secondly, Integrator forms a very intimate complex with protein phosphatase 2A. Now, this is a well-studied protein phosphatase that has many, many targets in the cell. But by binding to Integrator, it's brought onto chromatin, where it's proposed to oppose the kinases that activate transcription. So these points together suggest that Integrator is a negative transcription regulation factor, which tries to terminate transcription in the promoter proximal region. How do we get to know the function of Integrator at that detail that we wanted? First, we needed to purify the Integrator complex. And given the large size, this was quite a hard task. In the end, what worked was basically being able to identify the subcomplexes of Integrator that can be expressed together. And this allowed us to actually reconstitute the 17 subunit Integrator PP2A complex by purifying first the subcomplexes that we systematically identified. And on this is, for example, an SDS page where you see individual proteins like Integrator subunit 1, and then the rest of the subunit nicely distributed on this gel. Having the sample in hand, we can go ahead now to perform Cryo-Electron Microscopy and try to understand how Integrator works. Now, what is Cryo-EM? This is one of the Nobel Prize-winning techniques that has really advanced biology a lot in the past few years. And actually, most of the revolution in the resolution of Cryo-EM started when I was studying grad school. So I thought this was a perfect opportunity to delve into something new and probably be in one of the leading positions in the near future. Basically, the way this works is that you take your sample, which is this sample that we resolve on this gel, and rapidly freeze it in liquid ethane to make sure that the native nature, the nature of the protein is not destroyed. Basically, when you freeze things very fast, you don't form crystalline ice. We then expose those cryo-EM grids to very low dose of electrons at a dosage that the samples are not burned, but also provide enough contrast that we can see our individual particles as shown here. So beyond this point, we use a computer a lot, where we can actually identify individual particles, sort them in different views, discard broken particles, and finally make a 3-dimensional reconstruction. Because this signal is coming from interaction between electrons and the nuclei of atoms, we are able to align this, if everything works well, to high resolution enough that we can tell the position of individual atoms in this complex. And so we build a representation, what we call an atomic model, which tells us, on one hand, what the molecule we are looking at looks like in reality. On the other hand, we can also learn how it's formed. And on this slide, I don't want to go into the details of every interaction, but I just want to show that the structure we observed largely reflect what we figured out the hard way through biochemistry. In that, the complex can be grouped into 3 modules. The core module here, and then we have what we call the tail module, which is what runs around in the air up here, basically connecting to the core through the subunit 15, and then wrapping around the cleavage module, which is connected to the core with subunit 4, which is very nice and can also explain how our cells actually synthesize such molecules. The other thing that I want to highlight is that when we compare the fold of Integrator endonuclease to the CPSF73, which is the 3' processing machinery and endonuclease, it became clear again that even in the structural form, this is folded exactly the same, nearly identical, which then confirmed the initial hypothesis that Integrator is an endonuclease. One difference, though, was that when we compared the structure of Integrator in the structure to the structure of the CPSF that was trapped in an active conformation, it was clear that Integrator 11 in the free structure here is in a closed state, and I will elaborate more on this later. Additionally, PP2A forms a very intimate contact with integrator. But one thing that I want to highlight is that the active site of PP2A is blocked by integrator, suggesting that in the free form, integrator is completely inhibited. To understand how integrator works, we then went on to solve its structure in the context of a transcription elongation complex. And given that I'm running out of time, I will quickly rush through this part and ask me questions during the break. To understand how integrator works, we first ask whether it binds to the pulse elongation complex. And to cut a long story short, we form a pulse elongation complex here. We added a purified integrator, and then with this sample, we solved Cryo-EM structures. We were able to resolve two structures, one in the pre-termination state and one in the post-termination state. What this means is that we trap this complex before it actually starts pulling the complex apart, and then on the other hand, we were able to trap intermediate after it has pulled everything apart. What does this mean? We saw from the structures that integrator actually binds the pulse elongation complex by recognizing the pulsing factors as well as POR2, thereby being able to distinguish what a pulse elongation complex is from an active elongation complex. We saw that when integrator binds the pulse elongation complex, it immediately starts disassembling it. Using this tail module, it starts opening the so-called DSIF DNA clamp that I told you in the beginning locks the nucleic acids in the polymerase. Additionally, in the presence of POR2, the integrator endonuclease finally adopts an active conformation by opening the active site, which allows nucleic acids to enter the active site for cleavage. How can we be sure this is the case? We then went back to biochemistry to test this hypothesis that we got from structural prediction. And indeed, it became clear that the free integrator on its own is inactive, even though it's wild type. But once we give it a substrate, which is an RNA coming out of RNA polymerase II, it rapidly degrades it into oligonucleotides. This has a very huge implication because, well, first let's look at this movie before. This basically shows how integrator transitions from the closed state to an open state to bind the polymerase. And upon binding, we see that the tail module here starts opening the DSIF clamp, and then the nucleus is positioned very optimal, close to the exiting RNA. So RNA emerging from the polymerase directly proceeds into the endonuclease active site for cleavage. And then at the end, when the RNA is digested, basically the elongation factors that bind to POR2 only when RNA is coming out of the polymerase will dissociate. And so you're slowly beginning to pull apart this complex. Additionally, without RNA in the transcription bubble, the DNA template rewinds. And basically, the POR2 molecule is just loosely attached to this POR2 surface through the contact with the polymerase itself. So how do you disassemble this? That's what we explain using the post-termination structure. In this structure, one subunit of integrator actually binds in a location where it became clear that its presence prevents association with POR2. And this is easily appreciated in this movie. So you have the structure of POR2 and integrator, and its three basically binds between them, allowing the polymerase, preventing the polymerase from binding to integrator. At the end, integrator then returns to the free form and removes that subunit, and this state can bind polymerase again, which is what we show here in this cartoon, showing you how the free integrator is all folded up in a closed conformation, opens up to bind POR2, cleaves RNA, terminates, and its three, which is only loosely attached to the complex throughout all the other states, can prevent polymerase from binding to the integrator, basically recycling everything. And just to put everything back into context, at the promoter proximal region of genes, there is a key decision regulatory step which is based on this promoter proximal pulse intermediate. And so what really activates genes is the decision to either release such polymerases driven by kinases or to terminate it driven by integrator and other termination factors. And so with that, I will like to acknowledge my postdoc lab, Patrick Kramer, who has been really supportive and helped me also take on such exciting projects. And I learned from a lot of fantastic people during my time there. Most of them have also moved on now to take very fantastic places, so it's great to have such networks. As you work, try to be nice to your colleagues. And I also want to thank these very brave graduate students and postdocs who have been excited to join my lab here. And if there is time, I'm happy to take any questions. Yeah.